My name is John Sproul, and I'm at the University of Nebraska, Omaha. And first, I'd like to just acknowledge co-first authors on our preprint, Scott Hotaling and Jacqueline Heckenhauer, as well as our other fantastic authors, anchored by Paul Franson at BYU. And of course, I also want to just thank our funding sources for this project. Now, we know that repetitive DNA sequences comprise large fractions of most eukaryotic genomes. And we've moved past the paradigm of junk DNA, obviously, and we know that these repetitive sequences are integral to the composition, the structure, the function, and the evolution of genomes in many different interesting ways. And despite us having a growing foundation of repetitive elements, in most taxonomic groups, they remain understudied. And so we're in an exciting period where, with dropping costs of DNA sequencing and especially dramatic improvements in assembly qualities, where we can actually look at repeats in their context, it's an exciting time to be expanding to a more holistic view of how genomes evolve by including repetitive elements. So prior to this study, in a study led by Scott Hotaling that came out last year, we had assembled a group of 600 genome assemblies of 600 insect species. And we wanted to take that data set, dive into repetitive elements, and ask, what are the trends of abundance variation across clades? How often are repetitive elements impacting gene regions? What impacts do sequencing technology have on our ability to detect REs? And then we wanted to ask, if we take kind of a low-cost automated annotation approach, how good of a job is that going to do in a large taxonomically diverse data set? So I'm going to share just brief results from the last three of these research questions. First off, how are repetitive elements associating with genes across these clades of insects? And what we did is we took an approach that we put together as part of an earlier project led by Jacqueline Heckenhauer. And what we noticed is that often when transposable elements are immediately adjacent or even interrupting a BUSCO gene, the BUSCO algorithm notices open reading frames nearby, and it often just lumps those into its BUSCO annotation. And so by simply quantifying instances where repetitive elements are present in BUSCO genes, that actually gives us a standardized kind of surrogate measure for associations between repetitive elements and coding regions. So we applied that to our data set. And what we're looking at here are the various insect orders kind of in phylogenetic context. And each column represents the data from a given genome assembly or a given species. So what we found is that in this top pink track here, these associations between repetitive elements and coding regions evolve quite dynamically across clades. For example, in Hymenoptera, they tend to be quite rare, and we can contrast that with Trichoptera, Coleoptera, Lepidoptera, and other orders where they're surprisingly common. And in some cases, they impacted upwards of 25% of the genes that we surveyed. Now, we also noticed that those species that have a high proportion of these associations also tend to have high genomic proportions of lines or long interspersed nuclear elements, which are a type of transposable element. And so these findings, they put a spotlight on some potentially interesting insect lineages that could teach us a lot about the causes and consequences of repetitive elements when they're in and around coding regions, which is an interesting area of research. So we looked at effects of technology, and we found that overall, long-read assemblies showed a 36% increase in total repetitive elements detected. And that despite the fact that we did not see significant differences in assembly length in long versus short read assemblies. If we look at a few categories of repetitive elements, LTR showed by far the largest increase in detection in long-read assemblies at 162%. DNA transposons, lines, and other categories of repeats showed less respective enrichment in long-read assemblies. And so that helps us kind of have a baseline for what to expect in terms of technology-related bias as we're investigating different categories of repetitive elements in a biodiversity context. Again, we wanted to look at, if we just take an automated annotation approach, how good of a job is that doing in a large data set like this? And to get at that, we looked at the taxonomic representation of insects in the rep-based repository. So there are repositories like rep-base and DFAM that have repetitive element sequences, and software tools that annotate repetitive elements reference these repositories in order to assign a repetitive element in a new assembly to a given family of known repeats. And so we asked how many insect families of our 154 in this study have any representation in rep-base. And what we found is that about a third do, just 57. But a bigger story was that of those 57, very few are well represented with a lot of repetitive sequences in rep-base. And so all of these more or less white columns are effectively showing families that have just like 1 to 10 total sequences of repetitive elements in this repository. And what we'd expect is that this poor taxonomic representation of insects should have a big effect on how effectively we can, how well we can annotate repetitive elements in our assemblies. And one thing I just want to point out as we're here, these repositories, they really rely on community contributions, just like GenBank does. And so this poor representation isn't a poor reflection on the rep-based folks. It's really a reflection on the community and our best practices or lack of best practices in supporting these resources. So indeed, we found a big effect of annotation success related to this database representation issue. So here we've got of all repeats that we detected, what proportion are unclassified or were not able to be assigned to a known family of repeats. And as you can see in Drosophilids, an average of only 13% of repetitive elements were unclassified. The large majority could be assigned to a known family, whereas if we take all other insects and combine them, an average of over 40% were unclassified repetitive elements. And in some cases, upwards of 60, 70, 80% of repeats were unclassified. So this kind of large empirical data set, it really puts a big spotlight on the critical importance of taxonomic representation in these databases and how much we're limited in high resolution studies when most of what we're looking at in some cases are repeats that we can't identify to family. So you would hope that this is getting better as time goes on, right? As more genomes become available, these repositories are improving all the time. And while there is good momentum with some of these efforts, we did look at rep-base over time. And what we saw is that if we take how GenBank submissions, new assemblies belonging to previously unrepresented insect families in GenBank, we get a lot of increases in recent years, a lot of new taxonomic representation in GenBank assemblies, but we don't see that same trend in the blue bars, which is new taxonomic representation of insect families in a rep-base. So again, this is suggesting that community contributions are not keeping pace, even though we've got this big increase in genomes that are being published and made available. And so, this, I should point out, is an issue, this annotation bottleneck. This is a very well understood issue among circles of transposable element folks, right? This has been in the literature, in the TE literature for well over a decade, and it's often discussed. It seems, though, that there is an information gap or an awareness gap or a best practices gap when it comes to folks that aren't thinking about transposable elements and repetitive elements. And so there's really an opportunity to increase our awareness and embrace transposable elements, repetitive elements in our studies. And so if anything that I've said has piqued your interest, I've put together this slide with some next steps and some resources. There are several nice papers here that are really approachable that kind of lay out the landscape for us. Repetitive elements can really enhance what we know about our genomes. They can enhance our science. And so by embracing them and taking the necessary incremental steps to start to improve our ability to study them, we're going to get interesting biological insights right off the top. And we're going to set ourselves up to really be able to handle high resolution studies that keep pace with the excellent assembly qualities that are becoming available. And so I hope that you check out some of these resources and consider ways to continue expanding repetitive element literacy. And I appreciate the attention.